Hi everyone. Hi everyone. Welcome to the SESC Club this week. So I'm Hazel, and today with me we have Julie. We are both biologists here by tutoring. For the section of today, we will explore colorectal cancer plasticity, where you can see in the title of our seminar today. So we will have like a database-wide exploration of colorectal cancer metastasis with the main topic of progressive plasticity. Okay. And before we dive into this week's session, we'll first give you a little bit of recap of what we have presented last week. Oh, I'm sorry. Okay, so here we go.
Okay, so from last week, we looked at some IBD diseases, inflammatory bowel diseases, such as ulcerative colitis and Crohn's disease, and we were looking at bulk RNA-seq data. So here in this diagram, you can see we've broken down ulcerative colitis and Crohn's disease, and ulcerative colitis occurs everywhere in the GI tract, whereas Crohn's disease only occurs within the large intestine. We also found some of the epithelial cells in both the small intestine and large intestine are different in the disease than they are in the normal groups. You can see here through the bar charts. We also performed differential gene expression analysis on both ulcerative colitis versus normal and Crohn's disease versus normal, and we found some upregulated genes in both diseases. And then lastly, from there, we performed pathway enrichment analysis, where we found an enrichment of pathways related to immunity, immune response, and just the immune system in general. And this is what we expect, especially in a disease like inflammatory bowel disease, where there's a lot of inflammation occurring.
Yeah. So for last week, we use a smart bulk with bulk RNA-seq data. We explore IBD or one GI tract disease. We have performed, as we can see here, DG analysis, ANOVA analysis, pathway enrichment analysis, and then we also perform the deconvolution here. So you can rewatch this episode on our YouTube channel. And for this week, we still stay in the GI tract, but no longer IBD, but we actually explore colorectal cancer. And for today, we will use single-cell RNA-seq data. Okay. And okay. So this is the agenda of today. Before we dive in our findings, we first give you an overview of what is metastatic plasticity in colorectal cancer, and then we use our talk to data database to, let's say, investigate or explore the findings from an original article, and then we found four main findings here. So first, we found that the metastatic cells lose their identities during the metastasis process. Then we also found that tumor cells lose their gene correlation structure compared with non-tumor cell. And then after that, we go a little bit further into the metastatic process, where we found that metastatic cells undergo a non-canonical differentiation, and then they turn into some very abnormal cell types, which we will explore further. And then last but not least, we will show you one specific gene called ProX1, which has been shown to repress the non-canonical differentiation in colorectal cancer. Okay, and that's now we go into the first part, which we will explain to you a little bit about what is metastasis plasticity.
Yeah, so if you're not familiar with what metastasis is, it's the ability of tumor cells to change their state and behavior during metastasis to adapt with a new environment. Sorry, that was plasticity. So if we start from stem cells can differentiate into progenitor cells and then become differentiated cells. And then they can undergo other processes such as transdifferentiation and then dedifferentiation, where they become a differentiated cell into a progenitor cell, and so on and so forth. And so the diagram on the right, if we start in the colorectum here, our cells start here and they undergo metastasis, they can migrate into the liver and they can adopt behaviors and cell state changes within this liver to become more like liver cells.
Yeah. So about the plasticity here that we mentioned, let's say let me explain it in a very easy to understand way, that when the cancer cell moves to a new organs like liver in here, it will differentiate into, let's say, liver cell types instead of normal colon cell types. So that's what we call the plasticity, so that the cancer cell can adapt with new environment in new organs of the body. Okay. So as we mentioned earlier, the findings today we actually adopt from an original article called Progressive Plasticity During Colorectal Cancer Metastasis. This paper was published in Nature back in October 2024. And in this paper, the author actually, they collected samples from 3 different parts of the body. The first is the normal colon, and then they collect the sample from primary tumor, and then the metastasis size. So what very unique about this paper is that the author, they have, like they was able to collect 3 types of samples from one patient. Like they have multiple patients, and for each patient, they have 3 types of samples. So it eliminates that kind of like different sample effect. And then after collecting the sample, the author performed single-cell transcriptomic, multiplex imaging, and also they also have the patient-derived organoids to explore and to investigate the fate of metastasis cells in colorectal cancer. And beside that, you can see there's a heat map here showing, let's say, one of the most important finding in this paper, when they point out the metastatic cell actually underwent a process called non-canonical differentiation, and then they return into some abnormal cell type. To sum up the findings in this paper, we put it here, like there are 3 main cell states in the CRC, like the colorectal cancer progression. So first, the normal differentiated intestinal cells in the colon will transform into intestine stem cell or ISCs, like in primary tumor region. And then after that, these cells will undergo the developmental reprogramming to progenitor-like fetal state associated with epithelial injury, like associated with the stress in the body. And then in the third state, these progenitor-like cells will have like an expression of non-intestinal lineage gene programs during the metastasis process. So the non-intestinal lineage gene means that these cells will turn into, let's say, the squamous cell or different subtypes in liver cell. Yeah. Okay. And now, now that we have shown you the original paper, we will try to reinvestigate those findings in that paper using talk to data. Let's say we want to see the findings in a bigger picture on like a whole database wide. That is the main topic of today. And along the way, please let us know if you have any questions. Just type it down and we will answer it right away. Okay, now we dive into our first findings, which is metastasis cells lose their identities during metastasis process. Okay. Yeah.
Okay, so again, a reminder, here in the paper, the authors demonstrated how differentiated intestinal cells will become more stem cell-like in their primary tumor. So when we talk about metastatic cells losing their identities, we're talking about a decrease in intestinal stem cell programs in the colorectal cancer metastases. So these programs or these gene sets are decreased in our primary tumor.
Yeah. Okay. Okay, so we see that we see the heat map here showing the relative expression of some of the top differentially expressed genes across primary tumor site and metastasis site. And then you can see that the author actually grouped those cells into different color, representing for different like signature pathways related to ISC program. And the main, like the 3 main pathway, ISC pathway that the author mentioned was the Wnt signaling, the embryonic development genes, and also the stress response, which was shown to decrease in CRC metastasis. And actually, when we try to query the genes related to this pathway in TalkToData database, as you can see in the composition chart right here, that we got the condition of colorectal cancer, the sampling size we divide into site of metastasis and the primary tumor site. And then you see on the bubble heat map right here. So align with the findings from the paper, the database-wide findings actually also show that there is a decrease in ISC programs or ISC related genes inside of metastasis, as you can see here. So we also put down the average ranking or the expression level of each genes down here in this table. And today we also want to explain for you further a little bit about what is average ranking and why we use that in our database.
Yeah, so we represent gene expression by the average rank. So this operates under the assumption that each cell will express a maximum of 5,000 genes. And so if a gene were to express very highly in that cell will have a ranking closer to 5,000. Whereas if a gene were to express very lowly in a cell, its ranking would be closer to 1. So the reason why we represent gene expression by its ranking, this is how we can normalize the expression level across different data sets and combine data sets this way so that we can compare their gene expression levels.
Yeah, so Julie just mentioned about the assumption of 5,000 genes. Actually, based on the current technologies in single-cell RNA-seq data, the maximum level of detection is about 5,000 genes per cell. But in general, our cells will usually express around 1,000 to 2,000 genes in like today's technologies. Yeah. Okay, that's about the decreased ISC program in the metastasis. Okay, I'm sorry, let me go back here. Oh. Okay, now we go to the next findings and corresponding to our next question, which is how do the gene profiles of the tumor site and site of metastasis differ? So as you can see in this heat map right here, we only use some of the specific macro genes provided by the author of the papers, but now we want to, let's say, take a bigger, like a deeper look into the whole expression profile of the two sides, and then we hope that we can find something else besides some like ISC related programs. Okay.
Yeah, so basically we performed differential gene expression analysis and we found some upregulated genes in our site of metastasis and different upregulated genes in our primary tumor site. Right here, we're looking at our upregulated genes in our site of metastasis. And if we perform gene stent enrichment on these upregulated genes, we found pathways related to epithelial mesenchymal transition or EMT, which is a really important attribute of metastasis. We also found an upregulation of this WNT and beta-catenin pathway, which in this paper here is used as a biomarker for CRC. And in particular, we found that this gene, SMAD7, is associated with poor prognosis in colorectal cancer as well, according to previous literature.
Mhm.
Okay, and then in this next one, we're looking at the upregulated genes in our primary tumor site. And some of the important upregulated genes are WBSC-R22, which confers anti-tumor agent resistance in colorectal cancer. TUG1 promotes CRC metastasis via the EMT pathway, which we found earlier. And REG4 is associated with unfavorable prognosis and resistance to chemotherapy. So a lot of these upregulated genes are implicated in unfavorable prognosis of CRC.
Sure. Okay. So now we come to the second findings. Oh, the second part of this presentation. So in the first part, we actually explore the fate of metastatic cell. But now let's go back a little bit to the primary tumor size, where we found that the tumor cells also lose the gene correlation structure. So the gene correlation structure also related to the cell's identity. So this, let's say that in the primary tumor cell, in the primary tumor site, the tumor cells also lost their identities, and then which, like, let's say like an early prediction to the fact that they will eventually absolutely lost their identities and then move to like the metastatic state. Okay. Now in the paper, so this is the figure from the original paper, the author found that there is a weak within cell type gene correlation structure in cancer cells. So as you can see in this correlation heat map right here, on the bottom left, right here in the left part here, we got the untreated tumor or the tumor cells. And on the upper side here, we got the non-tumor. And then we have three main cell types in the colorectal cancer, which are like the ISC, the goblet cells, and the enterocytes. We can see very clear that in non-tumor cell, the correlation structure is very strong. It shows very clear that the genes of this group are correlated with the gene of that groups. But in the tumor, it's like faded away. And then we try to like validate that whether we can, but like on the whole database, whether this still happen or not. So yeah, the answer is yes. When we query for some ISC and enterocyte signature genes between the tumor and non-tumor region, we also can see that in the non-tumor region, the gene correlation structure is stronger compared to the tumor region. So again, validate the findings from the original paper. Okay. And then that is about the second part of this presentation, where we explore the fate of tumor cells in primary tumor site. And now we move to the next part, where we'll go deeper into, so what happened after the loss of identities in those cells. So what happened was those metastatic cells will undergo non-canonical differentiation. So non-canonical, let's say this is an abnormal way of differentiation. And then we'll explore it here.
Yeah, so when metastatic cells undergo non-canonical differentiation, this basically means that they are differentiating into a pathway which we don't expect. So they are differentiating into something that may be non-intestinal, for example. And we can validate this by looking at the modules that are corresponding to the canonical intestinal states, which are ISC-like, differentiated absorptive module and the secretory module. So from here, we hypothesize that the canonical intestinal states will decrease at the site of metastasis. And we can try to investigate this hypothesis using talk-to data.
Yeah. So in here, based on our hypothesis, we query for the genes related to canonical differentiation, which Julie just mentioned earlier, like the IC, like the differentiated absorptive and the secretory genes between the site of metastasis and the primary tumor site. And again, we also see that those genes related to canonical differentiation decrease its expression in metastasis sites compared to primary tumor. As you can see very clear that the genes expression are higher than in the site of metastasis. Yeah. Okay. And yeah, so after the non-canonical and canonical differentiation, so eventually we want to, let's say that, decrease cancer, like repress the cancer in patients, right? So the last but not least, findings from the original paper. So these genes was actually found in the original paper, the ProX1, was found to repress the non-canonical fate in the primary tumor site. So now I will explain for you further about the ProX1. So in the paper, the author found that this gene, ProX1, is highly expressed in the tumor front. As you can see, we have the staining right here in two different samples, and in primary tumor region here, you can see in the tumor front right here and right here, PROX1 in yellow color is highly expressed. So this expression is like the author hypothesized that the expression of PROX1 is like a gate to prevent the spreading of cancer cell. And why is the gate to prevent the spreading of cancer cell? That when the author investigate the correlation of PROX1 with other canonical module genes, they see that with the high expression of PROX1, the expression of non-canonical module actually decrease. It means that, yeah, PROX1 actually repress the non-canonical fate in the primary tumor site. However, once those cancer cells actually overcome the PROX1 gate, as you can see, like a very clear gate right here, they can now eventually turn into non-canonical differentiation, and then they can move to the metastasis site in the liver, as you can see here. And then in the metastasis site, we see that PROX1 is expressed somewhere here, but not somewhere here. You can see no yellow color here. And based on this finding, we also ask another question that, so does PROX1 expression inhibit a similar pattern at the site of metastasis? So in order to do that, we actually create two cell type, the two cell population. So in here you can see that we create two cell populations of colorectal cancer cells in metastasis site, right? CRC and metastasis. However, the expression of PROX1 is different. So in the first group, it will be PROX1 high expression, and in the second group, it will be PROX1 low expression. And then we query for the genes related to non-canonical differentiation. And interesting that we found the genes related to non-canonical differentiation are down-regulated in PROX1 high metastatic cell, which means that yes, the effect of PROX1 remain in the metastasis site. Okay, and just to let you know, like how we create this like two group of cells, we would like to give you a little bit of demo, because this is like a new feature on our platform. So we're really happy to show you guys around like how we created this. Okay, so okay, so we just mentioned that we use the colorectal cancer metastasis site, right? So first of all, on talk to data beta here, right here, first we want to create like a subset of, let's say, okay, let me query for here. Oh. Okay, so here we go. Colorectal cancer. Okay. And then we also want to filter out for the sampling site in the site of metastasis. Okay. Oh, let me see. Oh, oh, okay. I'm sorry, my mouse is going somewhere else. Okay, now we go to the site of metastasis. Okay. Okay, so if you have watched our talk to data comprehensive session before, you may use to this kind of like interface of where we actually filter our cells based on different ontologies like site of metastasis and the colorectal cancer. But today we want to also filter by the gene expression filter, which is the expression range filter right here of the PROX1. Okay. Okay. So, and then after we filter, we select the PROX1, we also go to, so this is the actual expression distribution of this gene across our database. So now because I want to, let's say, give it like a PROX1 low expression, right? I go all the way down to this low, and then I apply the gene expression right here. And after that, I can save this subset of cell as the CRC metastasis ProX1 low. Yeah, okay. And then I also I quite like the similar to make the two ProX1 high, where I just move the distribution to higher level. And then after that, when I have two subset of cells, and we use here, the term we use here is we have two experiments. And then we go to the composition chart, and let's say we select the CRC MET ProX1 high and CRC MET ProX1 low. And then, okay, let me go here. And then we delete this one. Okay. And then let's say we query for one gene that related to non-canonical differentiation, the EMP1, and let's say the L1CAM. Okay. Just use two of that, and then we apply. And that's how we create the composition chart. Okay. In here, we actually have, we received one question from Paramore that, okay, I have looked at your previous demo from the YouTube video, and my version looks different from what you show. Okay, so this is not about whether it's Mac or Safari, like or Chrome. Actually, this is like a new feature that we are going to release soon, maybe next week. And then the interface that we're using here is like in the testing phase, and it will be available next week. Yeah. So that's the update for Paramore. Okay. Okay. Thank you, Paramore, for your question. Okay, now we're done here. And yeah, that's how and also here, we also switch to the relative mode to view it better. Okay, and that's how we create our composition chart. Okay, now we can go back to the slide. Okay, and that's a little demo also ends our presentation today. Before we move on to the Q&A section, we would like to give you a summary of the section.
Yeah, so again, we reviewed the data from the original paper, and we kind of wanted to replicate and verify the findings database-wide. So that's what we did in this analysis today. We performed a database-wide investigation of colorectal cancer metastasis plasticity, and we demonstrated that we found similar findings to that original paper. One of the findings we found was that low expression of ISC-related genes was present in CRC metastasis. We also saw a loss of gene correlation structures within the tumor cells of CRC. Also, low expression of canonical related genes was present in CRC metastasis as well. And then finally, we found that ProX1 high cells in metastatic cells has low expression of these non-canonical related genes. So we found that ProX1 is repressing these non-canonical related genes in both the primary tumor site and in the site of metastasis as well.
Yeah. Okay, so that's all for the presentation today. Okay. Now on your screen, you may see that we are showing the survey again. So this survey is some questions that we really hope that you can fill in so that we can make talk to data beta version better. So we are in the developing phase of the talk to data beta version, and we would like to ask you some question about like what type of data or what type of features that you would like to be included in the platform. And yeah, so we will give you guys, let's say, 1 more minute to answer the survey. And along the way, please just let us know if you have any question. You can just fill in to the Q&A box. Yeah, thank you. Okay, so if we don't have any question today, we will end this session here, and we hope to see you again next week. Yeah. Bye.
Thank you, everyone.
Thank you for your attention.